30. So we will start our class. Okay, so to check your attendance, so you should enter 2039. 2039. Okay. Okay. 양예은. 양예은?
네. 네.
왜 지난 시간 안 나왔어?
준비 있는지 몰랐습니다.
정신 차려, 정신. 오케이.
네.
그러니까 조용은 아까 보이던데? 어디 갔지? 왜 결석이지?
예,
너 왜 출석 체크 안 했어, 지금?
지금 방금 막 했습니다.
오케이. 강지효 결석인데? 에너지화학공학과 이정길, 김지연, 최지연. 지난 시간에도 결석이고 오늘도 결석인데? Okay, Tibor, you have to enter 2039 to check your attendance, okay? Okay.
Yes, I will do it now.
Okay. 신진현.
네.
나노바이오?
네, 맞습니다.
왜 어제 안 나왔어? 아, 왜 어제가 아니고 왜 지난 시간에 안 나왔어?
저번 시간 까먹었습니다.
정신 차려. 오케이? 오케이. 오케이. Now we will start our class. Okay. So last class I explained the detailed information about the midterm exam. Okay. Okay. Do you have any question about midterm exam? So no question. Okay. Do you have any question about midterm exam? Okay. So if someone missed last class, so you should watch it, the last class. I already uploaded the file. Okay. You should watch it, the VOD. Okay. Okay, so once again, important thing is once again, so 21st, October 21st is the midterm exam. And also, I mentioned several important steps like this, okay? Okay, if you missed one of this step, okay, you will lose your point, okay? Okay, okay. Okay, so today we will cover chapter 4 and chapter 5, okay? Okay, so do you have any question in chapter 4 homework or the class? Do you have any question? So do you have any question? So maybe everybody already understand it, all of their material, course material. Okay, okay. So maybe you can cover chapter 4. So okay. So chapter 4, subject 1 was that amino acid composition and organization of the fibrous proteins. So also in chapter 4 homework number 1, so we have to describe the difference amino acid composition of collagen, silk, and keratins. Okay, so the important thing in the alpha keratins, so they have the higher cysteine value. Okay, cysteine content. Okay, cysteine is 11.2. Okay, how about silk? They don't have cysteine. How about collagen? They don't have cysteine. However, the alpha keratin, they have the 11.2% of the cysteine. And we also cover some important thing in the fibroin, silk, and also important thing in the collagen is like this. Okay. Okay, 강지호 좋아, 출석 좋아. And second thing is we cover the keratin. Okay, so the nail is the main, in the nail, the main component is the alpha keratin. Okay, so they are right-handed alpha helices. Okay, okay, and also this one, the repeating unit of the keratin structure is like this. We call it as the heptad repeat. Okay, important. Now I explain the important concept of the keratin. Okay, you have to know. Okay, you have to know. Okay, in our homework chapter for homework, we already covered the important step. For example, the keratin is based on 7 residue or heptad repeats. So describe the property of this sequence that the favor the cold structure. Okay, so for example, they have the heptad repeats. Okay, and their structure is like this. So okay, this one is A, B, C, D, E, F, G is like this, and also another strand is also A, B, C, D, E, F, G. Okay, but in actual midterm exam, you have to know how to draw the structure like this. Okay, A, B, C, D, E, F, G like this. Okay, and also especially in A and D location. So this is the hydrophobic residue is there. So usually the leucine or isoleucine, and valine or alanine is here. And also, how about the E and G? Especially in the E location, the glutamate is there. So glutamate is minus charge. How about G? And G is usually the arginine and lysine is there. So arginine and lysine is they have positive charges. So plus, minus, it means that they have the charges interaction between the E and G. Okay, so you have to know, okay? Okay, like this, okay. And also there is a 2 type 1 and type 2 keratins. So type 1 keratin, we call it as acidic keratins. So they have the more negatively charged side chains. For example, so usually so in negatively charged, for example, in E location, we have the glutamate and the negatively charged amino acids there. But in the C and B and F location, in the acidic keratin, they have a high content of the negatively charged amino acid. That's why we call it as a type 1 acidic keratin. So type 2 basic keratin is that usually in the G location, arginine and lysine, positively charged amino acids is there. And in addition to the G and C and B and F, this location, there are high content of the positively charged amino acids there. So that's why we call it as type 2 basic keratin. Okay. Okay, and also we cover some interaction stabilizing alpha keratin. Okay, and also disulfide bond is there, and ionic bond. Okay, okay. So and also hydrogen bonding is there, and also hydrophobic interaction between the A and D. There will be the hydrophobic interaction. Okay, and also fibroin. Okay, now we will cover fibroin. So in chapter 4 homework, we already there is another question. Okay, why is silk is both strong and flexible? Okay, so you have to know the reason. Okay, and also question number 2 is to explain why glycine and proline are found in high frequency in the triple helix of collagen. Okay, first of all, so silk fibroin consists of the anti-parallel beta strand, because they are the anti-parallel beta strands. So we already covered the anti-parallel beta strand. Each turn, from one pitch to another pitch, the distance is 0.68 nanometer. So it means that they are fully extended conformation. So in homework, there was one question regarding this one. Okay, so in homework, we covered that so why silk is more resistant to be stretched or shrink? Okay, because they are already fully extended conformation. So silk cannot be further stretched without breaking covalent structure in the beta strand. Okay. Okay, so this is the reason, and we already covered in homework. And also, their repeating unit is they have the glycine, serine, glycine, alanine, glycine, alanine. This is their repeating unit. So they have the glycine, serine, glycine, alanine. After that, they also have glycine, serine, something like that. So what is the characteristics of glycine, serine, and alanine? They have the small, the R group. For example, glycine, they only have H. Serine, alanine, they only have the methyl. And serine, they have finally they have the OH is like this, okay? Instead of H, they have OH is like this. Okay. And also they have the antiparallel beta strand, okay? So between the antiparallel beta strand, so they are the hydrophobic region, because nearly all of the amino acid, they are hydrophobic. So between this residue and this residue, they have the hydrogen bonding is available, and also there is the van der Waals interaction is available between the antiparallel beta strand. That's why they have the very flexible. They are very flexible. Okay. So they have this kind of the repeating unit, and between and also they have the antiparallel structures like this, antiparallel beta strand structure like this, and the antiparallel beta strand, they are hydrophobic because they are composed of the glycine, serine, glycine, alanine like this. Okay, that's why, and they have their van der Waals interaction is there. That's why they are very flexible. Okay. And also For example, and also they have the smallest side chains. Okay, glycine, alanine, serine. So they have the smallest side chain. These 3 amino acids consist of the 85% of the silk. So it means that they can induce close packing like this. For example, in this case, so interaction alanine and serinase is the interaction between is alanine serine happens. So around this the distance is that 0.57 nanometer. How about the other side? They only have the glycine residue. So glycine interaction, glycine and glycine is available. So their distance is only 0.35 nanometer. So it means that they can do close packing of antiferrous beta strand. That's why they are very strong. Okay, there's 2 reasons. Okay, we already covered here number 3 question. Okay, very important. Okay. How about the collagen? Now, the 4th subject is collagen. Okay, this is the major component. There is 2 types, type 1 and type 2. Okay, the major component is type 1. So they can see in the skin, tendon, they can see the tendon and the ligament and the bones like this. Okay, and also, and also their structure is they have the repeating unit. Okay, so each strand is this is N to C. They are left-handed. Each strand is, for example, this is also 1 strand. This strand is the left-handed, and this 3 strand is they combine together. This case is the N to C. Their 3 strand looks like the right-handed is like this. Okay, right-handed is like this. Okay, and there also repeating unit is the glycine, XAA and YAA. In XAA, proline, lysine can be there, and YAA, proline or lysine can be there. And also, especially proline and lysine, so usually the post-translational modification should usually happen like this. Okay. And also, this is collagen structure. And also, this is very important. Okay. In our homework number 2, explain why glycine and proline is found in high frequency in triple helix of collagen. The reason is why proline. Because proline, they don't have the amide hydrogen, they don't have H hydrogen. So this hydrogen, we call it as amide hydrogen. Okay, but the other in the peptide bond, every peptide bond, they have the NH group. This is the amide hydrogen. But they don't have, in proline case, they don't have amide hydrogen, especially in the proline cases. So it means that this absence of the amide hydrogen eliminates any potential hydrogen bonding. Okay, that's why this is to make the weak interaction. So weak interaction between the neighboring amino acid. So weak interaction between the strand. Okay, this is one reason. And the other reason is glycine. Glycine occurs every 3rd residue. So all of glycine is located near near the helix axis, like this. And also glycine only have, in our group of glycine, they only have H. So it means the minimal side chains allows the formation of the triple helix, it's like this. So they have the minimal side chain. That's why it can allow the formation of the triple helixes. If maybe, how about if the maybe tryptophan is located here, 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 maybe they cannot make the triple helixes. Tryptophan is very big, so they cannot easily make the triple helixes. But in the collagen, they have glycine. Okay, so this is the reason. Okay. Okay, so we also covered so interaction stabilizing collagen structure and van der Waals, and we cover like this. Okay, and also covalent bonding is also available in the collagen. For example, in the collagen biosynthesis, so final step, final post-translational modification is the covalent bonding happens. So they will make the lateral association of tropocollagen. So finally, they will make the fibril synthesis happen like this. Okay, especially in the collagen, so there is the covalent bonding is available. And also, so this is also in the collagen is the hydrogen bonding is available to stabilize the collagen structure between the amide hydrogen and the carbonyl oxygen. Between here and there is the hydrogen bonding is available. Okay, this is very important. Okay, as I mentioned earlier, post-translational modification is happening. So proline can become the hydroxyl proline. They have the OH is here, and also lysine can become the hydroxyl. Lysine is like this. Okay, that's why, for example, if there the repeating unit is a glycine proline, their transition midpoint temperature, their transition, so if there is a glycine proline, in this case, their transition midpoint temperature, okay, their transition around 50% around here, so their transition midpoint temperature is 17 centigrade. However, if they have the hydroxyproline is there, so it means that hydrogen bonding between the hydroxyproline and neighboring chains, it can make the triple helix stabilization. So this OH can make the triple helix stabilization. That's why their TM become the 37 centigrade. This is very important, okay? So you have to understand this procedure, okay? Okay, and also we cover in chapter 4, okay, homework, okay, 4 and 5. And also there is another question was like this. So why does the polyalprolines fail to form a triple helix? Why? Because they need to make the collagen triple helix, so they need glycine. So glycine should be occurs every third residue. However, polyalproline, they don't have the glycine, so it means that so polyalproline failed to form or triple helixes. Okay. Okay. How about will the sequence proline, glycine, proline, proline form or triple helix? Yes, but its stability is lower because they don't have any hydroxyl proline. Okay, but they are available, but the stability is they are a little bit unstable. Okay. Okay. Okay, and also this is also another evidence that the importance of the hydrogen bonding to stabilize the collagen structure. So collagen, they make the triple helix is like this. Okay, between the helix, they have the hydrogen bonding is available between the helix is like this. Okay, so if we add heating, make the gelatin make a disordered status is like this. After we cool the gelatin, so they will regenerate the triple helix. It's like very unorganized structure. That's why this is the gelatin. That's why the okay. For example, we had another question is like this. So gelatin is primarily derived from the collagen. The protein responsible for most of the remarkable strengths of the connective tissue. Collagen is the most remarkable strengths of the connective tissue in tendon and other tissue. So gelatin is usually soft and fluffy, and they also generally lacking the strength to explain this observation, because after we cool down, so they will regenerate triple helixes like this. So it means that this structure, actually this structure is very strong. That's why they can make the bone or they can make the ligament like this. But how about the gelatin? They are very soft because this is the region, okay? Okay. And also we cover collagen synthesis, biosynthesis. So in the red color was the post-translational modifications like this, okay? Okay, like this, okay? And also in our homework, there was a question about describe the post-translational modification of collagen and list these modifications in their order of occurrence. First of all, cleavage of signal peptide sequence is happen around the ER lumen is like this. Okay, that hydroxylation happen, so like something like that. Okay, okay. And also we cover some the genetic disease. Okay, for example, there are two diseases associated with collagen defect. First one is osteogenesis imperfect. In Korean, we call it as 골형성 부전증. Okay, so this is the bone, and this is bone from the osteogenesis imperfect patient. Okay, so if their symptom is severe, so they cannot stand like this because their bone is very fragile. Okay, their state, they look like this. Okay, the reason is that, okay, single base mutation, especially in glycine, it happens. So glycine can be single base mutation substitute glycine for another amino acid. It means that it can destroy the repeating unit. So finally, it will make the imperfect bone formation. So they will make the imperfect bone formation. So bone is very fragile like this. Another genetic disease is the Ehlers-Danlos syndrome. Okay, the patient who got the Ehlers-Danlos syndrome, their case is that they have the hyperextensive joint and also soft and elastic skin. So this one is also single mutation is happen in the nucleotide sequence encoding glycine, and they can destroy the repeating unit. Finally, they will have the hyperextensive joint and soft and elastic skin is like this. And very important, these two diseases is very important. Okay. Okay. And also we chapter 4 homework, we also cover these two diseases. Okay. Maybe in the, so we cover Ehlers-Danlos syndrome. Okay. Okay. And also, okay, we cover the number 10 is very important. Okay. 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 So do you have any question about chapter 4? So everybody understand it. Okay. 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 So, okay. How about we had better had 1 minute break because I need water. Okay. 1 minute break. Okay. 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 Thibault. So do you have any question?
No,
Okay.
not for the moment.
Okay. 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 It's good. Okay. 김준수, 메카트로닉스
네,
잘 이해하고 있어?
네, 4장이 제일 재밌었습니다.
오케이, 오케이. 열심히 공부하고.
네,
오케이. 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 뭐 간단하게 뭐 이야기하면 뭐 지금쯤 다 3학년 아니면 4학년이죠, 맞죠? 다 지금쯤 다 진로를 다 선택했죠, 맞죠? 지금부터 제일 중요한 거는 진로 선택에서 제일 중요한 거는 자기가 선택을 했으면 난 항상 그렇게 생각하거든요. 그 선택이 전 선택을 내리기 전에 엄청나게 많이 알아보고요. 내가 조사할 수 있는 건 다 조사해 보고요. 그다음에 이제 선택을 하고 나서는 절대로 후회하지 않아요. 후회를 하는 순간 이제 완전히 이렇게 망가지기 시작하는 거예요. 선택을 하고 최선을 다하면 여러분도 여러분이 원하시는 이렇게 여러분들 각자가 원하시는 바가 있을 텐데 다 이루실 수 있을 거라 생각해요. 충분히 포텐셜이 있고요. 여러분들 개개인 모두가 다 충분히 포텐셜이 있고요. 오케이. 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 Okay, let's go to the chapter 5. Okay. So do you have any question in chapter 5 homework or the class? Do you have any question? So do you have any question? Okay. Okay, let me go to the chapter 5. It covers the structure and function of the membrane proteins. Okay. First of all, we learned structure of the glycerol phospholipids. Okay, they have the phosphate. In this phosphate, there are lots of the several kinds of head group can be bind to the here. Also, they have another OH, another two arms like OH, OH. Okay, this is R1 and R2. Okay, so they will make the tail. Okay, so in C2, C1 and C2 position, the two long chain fatty acid can attach it to C1 and C2. Okay. So that's why their structure looks like this. Okay. Head group is they have the polar, and tail group they are non-polar. Okay. If there is cis double bonding is available, it means that they will make the cork is like this. So it means that their volume is increased. This one is affect transition temperature and also especially the fluidity. Okay. And also there is a characteristics of phospholipids. Okay. So phospholipid bilayer is consists of the two layer of phospholipids. Okay, with the hydrophilic exterior. So head group is hydrophilic, so they are go on the outside. And also non-polar region, they have the hydrophobic interiors like this. Okay, so this is electron density profile to show that their distance is around 4 nanometer. Okay, and also in chapter 5 homework, we already calculated the distance, okay, like this. Okay, this one. The question was like this. How many residue are required to span a lipid bilayer? And also we can calculate it like this. The lipid bilayer is 4 nanometer and something like this. So maybe like this, okay? Okay, so they need around the answer will be around like this, okay? Or something like that, okay? And also this is the determinant of phase transition temperature. Okay. First one is the very important. This is very important to solve the midterm exam. Okay. And this is the length of fatty acid chain. So if they have the short fatty acid chain, so it means that they are weak. They have the weak interaction. Because between their fatty acid chain and fatty acid chain, there is always van der Waals interaction is there. Because they only have H like this, so they are hydrophobic. So usually between hydrophobic location, there is a van der Waals interaction. So if their chain is the short, it means that short chain means that there is no there is not much van der Waals interaction, it means that they have highly fluidity, and means that they have a lower TM value. And also, if there is a cis double bond is available, so their structure is they will make the unsaturated R group is like this. Okay, if there is no cis double bond, okay, this is the lipid bilayer looks like this. So it means that they will have the unsaturated R group. It means that they will have the high fluidity. It means that they have the lower TM. How about the pH? pH is they have the high pH. It means that they have the loss of the OH minus, because the tail is fatty acid chain is they only have CH like this. If in the environment, if they have a loss of OH minus, it means that there will be the hydrogen bonding is available, and they will make this kind of the cork structure. They will make this structure like this. Okay, that's why. So high pH, they have the high fluidity. It means that they are lower TM. Okay, maybe you already know the determinant of phase transition temperature. Okay, very important. Okay, okay, and also we cover. Now this is second subject of membrane topology and function thin through the erythrocyte membranes like this. Okay. And also membrane domain is like this. Okay. Okay. Now is the we will cover band 3 protein. Okay. Maybe band 3 protein is located somewhere here. Okay, I cannot find it right now. Okay. Anyway, band 3 protein. They are integral membrane proteins. So band 3 proteins. Okay, they have the higher percentage of hydrophobic amino acid. So based on the hydropathy plot, so very important, okay, hydropathies group reflect the preference of amino acid side chain for polar and non-polar environment. Okay, if the value is higher like this, so it means that they are hydrophobic. Okay. So in this case, okay, I will show you it here. Okay, so in this case, okay, so in this case is the glycoporine proteins. This is also integral protein. Okay, based on hydropathies plot, they only have one peak is like this. Okay, so it means that they have one transmembrane domain. How about if they have the 2 peak? How about 2 peak? So it means that they have the 2 transmembrane domain. So you can make the 1 transmembrane domain, you can make the 2 transmembrane domain is like this. Okay, maybe each transmembrane is connected by another proteins like this. Okay, and also cytoplasmic C-terminal domain, if they have hydrophilic amino acid is like this. Okay, so in actual exam, so I will show you the hydrophobic, I will show you the hydropathy plot. Okay, so based on the peak, so you can draw the picture of the integral protein. So if they have 2 peak, so they are the membrane protein. there the membrane protein will be 2. If they have 3, they will have 3. They have 4, they have something like this, okay? Okay, like this, very important, okay? Okay, and also we cover the external, for example, glycoprotein A, they have external and terminal domain. They have external and terminal domain, they have highly hydrophilic amino acids, especially the inserine, threonine, and asparagine. For example, serine and threonine, they have OH group. So in this case, is the O linkage happens. They have OH group, OH group, OH group. So in this case, is O linkage glycosylation is happening. How about the asparagine? So they have the N group. So in this case, N linkage happens. So in our homework number 1, we cover this one, okay? So explain the post-translational modification added to the integral protein, especially in glycoprotein A. So they have O-linkage glycosylation, and also they have the N-linkage glycosylation, okay? And also, very important, this one, because they only have the one peak, so it means the membrane domain, they only have one membrane domain. If they have 2, they will have 2 membrane protein. If they have 3, they have 3 membrane domain. Very important. Okay. And also we cover that usually when we study the membrane protein, so we always used erythrocyte because we have the reason. They have the advantage, disadvantage, and also in our homework, we cover this one. Okay, so describe the advantage and disadvantage of the erythrocyte membrane as a model for studying membrane protein structure and organization. Okay. 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 So, and also now we will go to the bacterial rhodopsin. So they have the 7 transmembrane helices like this. For example, if they have 7 transmembrane membrane, it means that when we do the hydropathy plot, maybe they will have 7 peak like this, okay? Okay, so in the transmembrane helices, they have the high content of the hydrophobic amino acid around, for example, alanine, leucine, isoleucine, and valine. Okay, in our homework, we also cover this issue. Okay, so I will show you, we can show you this kind of structure. So based on the structure, based on this amino acid, so we can find that the region which has a higher content of the alanine, leucine, isoleucine, okay, something like that, okay? Okay, so we can cover, so this region may be the transmembrane domain because they have the highly content of alanine, leucine, isoleucine, valine. Okay, one in actual exam, so you can, so okay, this is answer, but some case if you did it, your answer, even though your answer should be, your answer is like this, it's okay. Okay, this one is also okay. But if you defined around this region, so you are wrong. Okay, your answer is not right. Okay, 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 and also bacterial rhodopsin. They have 7. How many? 7. Important 7 transmembrane domain. Okay. And the loop region, they have charged highly content of charged amino acid, for example, aspartate, glutamate, or lysine, or arginine. And also we cover the bacterial rhodopsin mechanism. If the light is absorption, it happens. Okay, so there is a retinal structure will be changed. So they will make the all transform isomer will change it to 13 cis retinal. And so this one you change it to this one. So it means that the H plus can be transported from cytoplasm to the extracellular spaces. Okay, after that they can recognize the light. Okay, so this is bacterial rhodopsin. We cover this one in homework. So for example here. So this one is very important. Explain the determinant of phase transition temperature. Okay, and also briefly explain the mechanism after bacterial rhodopsin absorb the light. Okay, and this is bacterial rhodopsin. They have a 7 transmembrane, and also another one, rhodopsin. Is also they have composed of 7 transmembrane helices. Okay, and for example, another question. Actually, there was a typo, but this is the rhodopsin, not bacterial rhodopsin. So explain the signal pathway that takes place in transducing after rhodopsin absorb the light. Okay, so this is the answer. Transducing is that they are heteromeric G protein. They are composed of the alpha, beta, gamma subunit. So this is the mechanism. It's like this. Okay. Okay. And also beta-2 adrenergic receptor. They also have 7 transmembrane domain. Okay, their function is like this. Okay, and also we cover photosynthetic complex. So now we go to the structure of bacterial reaction center. Okay, first of all, we learned the LH1 and LH2. They look like this. And also, especially the bacterial reaction center. So this is the reaction center. So this is the electron transport system is available. So this is their electron transport pathway in the reaction center. It looks like this. Okay. And also they also have cytochrome B1, BC1 complex is like this. Okay. So finally, they can use the electrochemical gradients. Okay. So they can make the ATP is like this. Okay. Okay. But the important thing, okay, in our homework, we cover explain the movement of electron in the reaction center. Okay, from the bacterial chlorophyll dimer and to bacterial piopitin, after QA and QB is like this. Okay. Okay. And also in the cytochrome and the cytochrome B1, BC1 complex. So we can see that they have some information is like this. Okay, but this is not important. Okay. Okay, and also we learned about their reaction center structure, especially the L subunit. This is L subunit is like this. This is a reaction center. So this is the reaction center, and this is the structure of their reaction center because they have the transmembrane domains. H subunit and cytochrome subunit, they don't have transmembrane domain. Cytochrome domain, maybe they have one transmembrane domain, I think. Okay, but the important thing is L subunit and M subunit. Okay, because they are composed of the. Okay, so L and M subunit, they have the two-pole symmetries like this. So they have the A, B, C, D, E. They have 5 transmembrane helices. Okay, 1, 2, 3, 4, 5. Okay, how about this one? How about bacterial rhodopsin? They have 7. How about in the reaction center? L and M subunit, each one they have 5 subunits, 5 transmembrane helices. And also we cover in the homework, we cover that there was a question. So why is the difference between each other? Because especially in the E subunit, helix E is they have the kink structure like this, caused by around here they have the proline. So they have the kink caused by a proline residue. That's why they have the difference between the actual length and estimated length. So I just write down here, I just calculate the estimated length, but the actual length is different because especially the E, helix E, they have a cork like they have a kink like this. Okay. Also, so cytochrome subunit, they don't have transmembrane domain, but they have 9 helices, 1, 2, 3, 4, 9. Okay, and their function is like this. And also, between an L and the M subunit, there is the active component is located here. This is active component. Okay, they are two-pole symmetric structure. Okay, and this is also the electron is start from the bacterial chlorophyll dimer, and they go to the bacterial phenol, PO15. They go to the QA, and QB is like this. Okay, so we cover this one. We cover this one. So this one is just starting from the question was that explain the movement of electron from Hem group in the cytochrome subunit to the active components present between the L and M subunit in the reaction center. Okay, from the Hem group. In cytochrome subunit, they transport 2 electrons to bacterial chlorophyll dimer. After that, these 2 bacterial chlorophyll dimer transport 2 electrons to bacterial PO15. And that QA, QB is like this. Okay. Now, okay, this is not important, okay. Now we will go to the photosystem 2, okay. Okay, now we go to the oxygenic biosynthesis. So oxygenic biosynthesis, for example, this one is located in the bacteria. This reaction center and LH1 and LH2 is located in some bacteria which they can photosynthesis. But usually the oxygenic photosynthetic system is present from algae and plants. They are composed of photosystem 2 and 1, like this. And this is the 2 structure. And also, finally, we will cover 1 structure is like this. Now we are covering the transmembrane domain, especially D1 and D2. They have how many? 1, 2, 3, 4, 5, and 1, 2, 3, 4, 5, similar to L and M serine. Okay, and also they have another transmembrane domain, CP47 and CP43. They have 6 transmembrane domain. Okay, and also photosystem 1 looks like photosystem 2. Okay, so instead of the D2 and CP47, they have a PSA8. So they have the 11. 1 to 11 is important. 11 transmembrane helices. Okay, 5 is the their C-terminal location is located nearly similar to D1 and D2. And the N-terminal looks like the antenna, CP47 and CP43 function as an antenna. Okay, they're similar to the CP43, CP47 and CP43 is like this. Okay. Okay. And the final one is the OMF purine. Okay. So their structure looks like the better, like this. So they have 16. They have a 16 beta strand structure. So they have a 16 beta strand structure. So they look like the better like this. Okay. So so in this region, they have a blue is amino acid, which is the hydrophobic amino acid. So asymmetric distribution of hydrophobic acid is available. So this is the contact region with the lipid bilayer. How about in the cavity? So they have the loss of hydrophilic amino acid is like this. That's why they can facilitate the entry of small polar molecule into the periplasmic space is like this. Okay, purine is composed of the which structural feature? Beta barrel. Okay. Okay, and also we cover final question was, so explain why OMFF purine can facilitate the entry of a small polar molecule into the plasmic space, because the cavity is composed of the hydrophilic amino acid. Okay. Why OMF purine can be contact with the lipid bilayer? Because in the contact region, they have the asymmetric distribution of hydrophobic amino acid. Okay, so now we covered all of the question. Maybe I think all of the question. Okay. Okay. So do you have any question about chapter 5? Do you have any other question? 권정봉, 다 이해했어? 이해했어?
네,
군대 갔다 와갖고 힘들 텐데.
열심히 하겠습니다.
Okay. Okay. So I already explained very important thing in chapter 4 and chapter 5 already. Okay. Okay. Very important thing. Okay. So now you can study. Okay. Okay. You can memorize. Okay. Okay. Is there any other question? I found that some of the people 에너지화학공학과 이정길, 김지연, 최지현 얘들은 지난 시간에도 안 나오고 오늘도 안 나왔네. Okay. Okay, do you have any other question in the chapter 2, chapter 3, and chapter 4 and chapter 5? Do you have any other question?
교수님,
Okay.
혹시 5단원의
응.
과제 중에 3번 문제 다시 한 번만 설명해 주실 수
오케이.
있나요?
The question was that explain the number 3, okay? So find the membrane spanning region. It means that membrane spanning region, most of the component will be the hydrophobic amino acid, most of, but not all of them, most of them, especially, for example, the aliphatic, which the amino acid which had aliphatic side chain, for example, isoleucine, and leucine, and alanine, and the valine. Okay. For example, okay, you can find that E is not hydrophobic, and also E is not hydrophobic. Okay, S is a little bit difficult to say something, but isoleucine is that they have the aliphatic side chain. Okay, for example, and they don't have, in this region, they don't have any minus charge, nor they don't have any positive charges. You find the valine, alanine, leucine, isoleucine. So maybe this region, the red region, we can assume that red region will be the membrane spanning region. Okay, but in the so that's why I said that the red region can be interpreted approximately as the membrane spanning region. However, your answer should not be exactly the same as the indicated region. So you can answer it like this, or you can answer it like this. However, maybe if your answer is like this, you are wrong. If your answer is like this, you are wrong. Okay? Okay. Is there any other question?
감사합니다.
Yes. Is there any other question? Number chapter 2, chapter 3, and chapter 4, chapter 5? Okay. And good luck in your midterm exam. Okay. Okay. See you next Thursday. Okay. It's right. See you next Thursday. Okay. Let's bye.
감사합니다.
Okay.
감사합니다.
감사합니다.
Okay.
감사합니다.
Okay.